Hello, thank you for inviting me to this conference. I am David Cross, geneticist at CIRAD. I work on perennial crops, mainly on oil palm and rubber tree. My talk is about genomic selection. I will first give some context, then I will present the principles of genomic selection, and I will go on with an example taken from oil palm, a species where the application of this method started a few years ago. And I will finish with some trends for researches on genomic selection. There are many plant-based products, including food, fiber, and so on. There is an increasing demand and at the same time a strong need for the sustainable intensification of agriculture that is producing more on already cultivated land while reducing environmental impacts. The agricultural system also needs to adapt to climate change. So in this context, plant breeding has a key role to play. Perennial crops, including coconut, are characterized by long life cycles, late reproductive stages, and extended evaluation period in the field. This makes that for this species there are specific breeding challenges and constraints. At the same time, these species are the ones that will benefit the most from recent scientific and technological advances, that is next generation sequencing, high-throughput multi-omics data acquisition, high-throughput phenotyping, and new biostatistical modeling approaches that combine high-dimensional multi-omics and environmental data like artificial intelligence. In this presentation, I will only consider quantitative traits, that is, traits controlled by many genes of small effects, which makes that there is no obvious way to classify individuals in a limited set of distinct phenotypic categories. For the genetic improvement of quantitative traits, we must consider the rate of genetic progress, which is given by the breeder's equation. It relates annual genetic progress, that you can see here, with different parameters. First one is the selection intensity here, which indicates the stringency of the selection progress, and it's also related to the size of the population. Here we have selection accuracy, which indicates the reliability of the estimates we have regarding the genetic value of our plant material. And here we have generation interval, which is the number of years required to make one breeding cycle. In perennial crops, we usually have long breeding cycles, which is detrimental for the rate of genetic progress. We also have usually a reduced number of individuals evaluated in the field, so that is reduced selection intensity, which is also detrimental for the rate of genetic progress. However, it is possible to have a good selection accuracy, for example, when field trials correspond to progeny tests or clonal trials. This is the case with oil palm, where selection accuracy of field trials is around 0.9. In these conditions, what about marker-assisted selection? If we can predict genetic values simply from profiles of DNA markers, then we could increase selection intensity, and we could make breeding faster. The change in selection intensity or generation interval would mostly depend on practical choices made by the breeders. I will explain later the case of selection accuracy, which is more complex and central in the way marker-assisted selection can be applied. Many studies were made regarding the detection and use of QTLs. These methods are very efficient at identifying genes and alleles that control the traits of interest. But this only captures a part of the genetic variation, which is usually not enough for practical breeding. The main problem for quantitative traits is that there is a large number of QTLs with small effects which are not detected, while they weigh a lot in the genetic variation, because there are many. That's how we end up with genomic selection. The first study on genomic selection was a simulation study considering quantitative traits for cattle breeding in 2001. The authors estimated the effect of around 50,000 SNP-based haploblocks from a set of individuals with phenotypes and genotypes. They measured the efficiency of the method considering the prediction accuracy, which is the correlation between predicted genetic values and true genetic values. I already mentioned this in the breeder's equation, but to give greater details, it can go from -1 to 1. And here, you have example plots with increasing levels of accuracy, going from 0, which is equivalent to random selection, to an accuracy of 1, where genetic values are perfectly estimated. In their study, they obtained accuracy of 0.85, which is very high. So they concluded that genomic selection had a great potential for the genetic improvement of quantitative traits. Today, it is the most common method of marker-assisted selection for this type of traits. It is now widely implemented in animal breeding, in particular in dairy cattle, but also in beef cattle, pig, fishes, and so on. This was made with a massive effort in terms of genotyping. For example, in dairy cattle, there were millions of cows genotyped worldwide, and most of this genotyping was done using SNP arrays with from 40,000 to 80,000 SNPs. There are now reports showing that genomic selection more than doubled the rate of the genetic progress in dairy cattle, in various dairy cattle breeds and countries. You can see it here with the example of the Canadian Holstein dairy cattle, where you see the trend in the performance before and after the introduction of genomic predictions for two key traits indicating the genetic potential for lifetime profitability and performance. In plants, there are more and more examples of the application of genomic selection, in particular by private companies. You can see here Limagrin, which reports that they now apply genomic selection to select the most promising plants early in the selection process. In these other breeding companies, they also use genomic selection. They mention that on their website. They use it to improve selection accuracy and to go faster in the breeding process. At Syngenta, they indicate that for complex traits, they use genomic selection. Now, let's see how genomic prediction works. So first, you have a set of individuals that are both phenotyped and genotyped, so they can be individuals or lines or crosses. They are genotyped with a dense covering of the whole genome, so they make the training set. In the second step, you use a statistical model, some statistical model, where you consider jointly all the markers. So there are two broad types of approaches. So the first one consists in estimating an effect associated to each marker. So it's the case, for example, of the statistical approaches called RR-BLUB or the Bayesian alphabet. Another type of approach, which is usually equivalent, consists in using the molecular markers to compute genomic relationship matrices between the individuals. Then, once you have this model optimized for your population, then you use it to predict the genetic value. So it can be the breeding value or the total genetic value, that is including, in addition to the additive effects, including also the non-additive effects. So you predict these genetic values for your population of selection, your selection population, comprising individuals that must be related to the training ones. So from that, you obtain the genomic estimated genetic values, and then you use that to make selection. Here you have an example of a genomic breeding scheme. This example is taken from a perennial crop, eucalyptus. So it starts with a population, which corresponds to the training population. So that's trees that are phenotyped and genotyped. So you use this population or different trials in different sites and so on to validate the GS model. So that means identifying the best model, because we'll see there are different types of statistical approaches. You can also vary the type of molecular data, the marker density, and so on. So you have to evaluate that at this stage. So once you have an optimized model, you know it's efficient, you know the accuracy for your traits of interest, then you can apply it on a population of individuals. So typically that can be young individuals. So here you see it's seedling candidates. So you predict their genetic value. Again, a total genetic value, if the idea is to release, let's say, varieties as a clonal material, or you can just predict the breeding value if the point of these individuals is to use them, for example, to generate a new generation, a new population that will be used in a type of recurrent genomic selection breeding scheme. Now let's talk about the accuracy of genomic selection. So again, it's the correlation between the genomic estimated genetic values and the true genetic values. It's the Pearson coefficient of correlation. It's affected by a large number of different parameters, so that's why there are so many studies, in particular at the beginning of genomic selection. That's why there are so many studies about the accuracy of genomic selection. It's affected by the trait, in particular because the heritability of the trait has a strong effect on the accuracy. It's also affected by different parameters related to, let's say, the marker data. So it's affected by the linkage disequilibrium between markers and genes. So this is related to the marker density and also to the number of genes involved. It's also affected by the type of markers, even if today we mostly use SNP markers. Selection accuracy is affected by the population size for the training set. The larger the training set, the greater the selection accuracy. Accuracy is also affected by the type of model or the type of statistical approaches we use. So it can be linear mixed model with BLUP approaches or Bayesian approaches, or it can be AI tools. Accuracy is also affected by the relatedness between the training and the test individuals or the application individuals. So the higher the relationship, the greater the selection accuracy. The accuracy of genomic selection can be estimated using real data, so experimental data you have from your breeding scheme. So typically it requires to have two sets of individuals or lines and so on with data records. So they must have phenotypes or any kind of estimates of their genetic value and also a genotype. So you need two sets, and one set is used as a training set. So you will train the model using their genotypes and their data records. And the other set is used as a validation set. So you'll make predictions of the genetic values for these individuals using their genotypes. And finally, because you also have data records on the validation set, then you will use that to compute the prediction accuracy in the validation set. So it will be the Pearson coefficient of correlation between the estimates of genetic values and the data records. So it's not exactly the same as the selection accuracy, because here you use the data records as reference values and not the true genetic values. Let's see now a model that can be used to estimate breeding values for a set of individuals. So this is the most simple example of model for genomic predictions. So it will estimate breeding values, not total genetic values. In this model, you have here a vector of phenotypic values, data records, and here different terms, which makes that in this model, we consider that each phenotypic value is equal to the mean value of the population plus a genetic term associated to the marker effects, plus the residuals. So here we have the Z matrix, which is an incidence matrix, which is filled with the genotypes of the training individuals. And here M is a vector of marker effects. So that's what we are interested in. So once you have analyzed this model, then you have an estimate of the marker effects. So this is the M hat vector. Once you have that, you just have to use another matrix of genotype, the matrix for the selection individuals, and then you will compute in a quite simple way, just making the product between the matrix of genotypes of the test individuals and the vector of marker effects, just making this product, then you will get an estimate of the breeding values of your selection candidates. So to see that in greater details, you start with a matrix of SNP markers. Let's say it's the M matrix. So you have individuals in rows here. You have SNP markers in columns, and this is filled with values that can be either 0, 1, or 2, which indicates the number of copies of the reference allele for each marker. Then, in the M vector, you will have one element corresponding to one SNP. Then, from this M matrix of genotypes, there is this small, simple adjustment to make that uses the allelic frequencies. So here you have the M matrix. Here you have a similar matrix with individuals, SNPs. And for each column, you have twice the frequency of the reference allele of the SNP. You apply this simple formula, then you get what is called the Z matrix. So it's genotypes, but they were adjusted based on the allelic frequencies. The point of that is that if you remember, for a given locus, a given gene, the additive value is given by this formula. You have one formula for each genotype, and this formula actually uses the number of copies of the reference allele, 0, 1, and 2, but it's adjusted by the frequency, the allele frequency of the reference allele. So once you have the Z matrix computed this way, what you have actually exactly matches with the formula we have in quantitative genetics. So that makes that what you estimate in the M vector is actually the alpha value from quantitative genetics textbook, which is called the substitution effect of the marker. So from that, you can compute the genomic estimated breeding values simply by multiplying each line, each row in the Z matrix with the vector column with the alpha values of the SNPs. So now how to solve the model I just presented. So it's actually a mixed model. So here we consider that the vector of marker effects follow a normal distribution with a specific variance. So this is the variance of the substitution effects of the markers. So there are different ways to solve this model. A very common one is to use the BLUP methodology, in particular the RR-BLUP. This will give you the M hat vector with the marker effect. There is an equivalent model where, in this case, instead of having the ZM term, you actually have something simpler. You have here the vector J, which actually contains the additive value of your individuals. So this vector follows a normal distribution, but in this case, it involves a matrix. So this term is a matrix of covariance. So it takes this J matrix, which is actually a matrix of realized or genomic relationships computed from the marker data. So this can be solved again by the BLUP methodology, and when you proceed like that, you are applying the JBLUP method. So usually RR-BLUP and JBLUP are equivalent, but in the case of JBLUP, the computation is actually easier and faster. It takes less memory. So this is actually the most common method used for genomic predictions. There are many ways to compute the J matrix, so I'm not going into details about that here. There are many other approaches that what I presented. I presented the additive RR-BLUP and the additive J-BLUP. It's obviously possible to model non-additive effects, dominance or epistasy, and other mathematical approaches are possible other than the BLUP, so Bayesian methods, machine learning like RKHS or neural networks. So the point of these different methods is to try to match with the diversity of the expected genetic architectures, depending on the distribution of the gene effect, if it follows a normal distribution or not, depending on the level of interactions between genes, if you have a lot of non-additive effects, for example. Let's see now another key point of genomic predictions. So it's the genotyping part, how to acquire the genotypic data. So genomic predictions require high-throughput genotyping. Before we have to genotype a large number of individuals with a lot of markers that will cover the whole genome, all the chromosomes. There are two major methods for genotyping for genomic predictions. One is genotyping by sequencing. So I guess you know well this method. It relies on the use of restriction enzymes, and it allows sequencing just a fraction of the genome. So GBS is good because it's rather easy to implement, it's rather cheap, but the drawback is that there is a high rate of missing data and a rather high rate of errors in genotypes. So the other major option is the use of SNP array. So it gives a better data quality. So the SNP arrays are developed through sequencing and resequencing of appropriate individuals. They have to cover all the genetic diversity corresponding to the population where you want to apply genomic predictions. It's also interesting because it gives the opportunity to include results from other studies. So for example, if you have SNPs in candidate genes, you can include them in the SNP array. And there are different formats you can see here, going from 24 individuals, 24 samples per plate, up to 384. Once the molecular data have been obtained, there is a final step regarding them. You need to impute the missing data. So usually it's sporadic missing data, so the genotype data that are missing because something went wrong in the genotyping process. So it's mandatory because the GS models, they require complete data sets of molecular data. They cannot handle missing data, even if it's just a few, like with SNP arrays. It's also useful when you want to combine molecular data obtained in some individuals with high density technology and for other individuals with a lower density method, which is done sometimes for cost reasons, to decrease cost. There are many software to do that. You have examples here. I think Beagle is the most common. That's the one we use in our studies on oil palm and in rubber tree. It's very efficient and very fast. So you have an example here about how the approaches, the imputation approaches can proceed. So in this example, it's imputation based on linkage disequilibrium between markers. It takes data from a set of reference individuals here that were genotyped at high density. So typically these individuals can be the founder or ancestors of your population you want to use for genomic selection. And then here you have a second data set of individuals that can be the selection individuals that were, for example, that were genotyped at a lower density. And then the method, the imputation method, will consider this data together and use the LD pattern to fill the missing data here with the most likely data. So let's go now to the third part of my talk, which is an example of the use of genomic predictions in a perennial crop, so oil palm, where we do hybrid breeding. I start with a few words about the species oil palm. So it's the first oil crop in the world, with a world production which is now above 75 million tons per year, with an expected demand around 2050, which could be above 120 million tons per year. There are various quantitative traits of interest in oil palm. Oil yield is the major one, oil yield and its components, so number of bunches, average weight of bunches, and so on. Resistance to diseases, that's also quantitative traits. There are three major diseases in oil palm. Vegetative growth, that's also quantitative traits. Conventional breeding is efficient in oil palms, so that's what you can see on this plot. It starts in the 60s and it shows the yield. So the increase here is only related to genetic effects. It's not related to agronomic improvements. There are two colors depending on the environment. So favorable environment for oil palm in Indonesia and less favorable environment in Côte d'Ivoire. In both cases, the increase is the same. Here it's 60% increase in terms of oil yield in 60 years, so it's 1% per year genetic improvement, which is actually quite good. Regarding the biology of the species, oil palm is an allogamous species, so they are male and female inflorescences on the same individual, and they start making flowers, inflorescences, at 3 to 4 years old, depending on the genetic of the plants and the environment. A very good point for breeding is that artificial pollinations are easy in oil palm, quite easy, because the inflorescences are big, so they are easy to isolate. They make a large number of seeds, possibly thousands. The pollen can be stored for years in a freezer, so that's very convenient for controlled pollinations. The drawback is that vegetative multiplication is difficult, so it's possible, but in the lab, there is no natural way of vegetative multiplication in oil palm. So it's possible in the lab, but it's quite complex, technical, so it's expensive, it takes quite a lot of time. So it's not really used for breeding. And now I explain an important point in oil palm breeding. So that's the results of an experiment implemented in the 60s. So there are different populations in oil palm. So Delhi from Southeast Asia, Lamé from Côte d'Ivoire, for example. And in the 60s, they made crosses between the different populations, and they obtained this result. So here you have the number of bunches or the average weight of the bunches. So here you see that in the Delhi population, you have a small number of bunches, but they are big. And for the Lamé population, it's the opposite. So you have a rather large number of bunches of small size. And when you check the hybrid between them, they are like average for these two traits. So these two traits, they behave like mostly additive traits. But when you check the total bunch weight, which is the product of these two base traits, then the Delhi and Lamé, they have similar total bunch weight, total bunch production, but the hybrid display quite high level of heterosis, hybrid vigor, they recorded more than 20% increase in terms of bunch yield. So this led to the definition of two heterotic groups called A and B, and central to the breeding of oil palms. So the breeders, they adopted a breeding scheme called the reciprocal recurrent selection. So you have the A group and the B group. They are bred separately, but evaluated one against the other. One cycle lasts around 20 years, and it's mostly due to the length of the phenotypic evaluation here. So here it's actually progeny tests. So in the fields, we have trials with typically hundreds of hybrid crosses that are evaluated for 10 years. So that's what makes most of the length of the breeding cycle. And here, within each group, the individuals are selected based on their combining ability, so on the additive part, so it's the general combining ability, and also on the specific combining ability. Now, to implement genomic selection, it works like that. We have a set of individuals that are all phenotyped and genotyped at high density. So we used these designs to test different prediction models. So we can vary marker density, the type of statistical method, and so on. And once we identify good models for a given trait, then we can apply it on a population. So typically it can be young individuals at the nursery stage, so they are the selection candidates, and we can predict their breeding value or total genetic value. In oil palm, many studies were implemented on genomic selection. The first one was in 2008. It was a simulation study where the authors, they compared two schemes for oil palm. So the first one was a phenotypic scheme here. So with cycles that takes around 20 years to complete. And here they considered an alternative breeding scheme where after the first cycle, they used the data to train a model of genomic predictions that was used for the following cycles, which allowed them to consider that during 37 years, they could make a total of 4 breeding cycles. Here are the results they obtained. So because it's a simulation study, they had to consider 20 and 80 QTLs because we don't know the actual number. And they presented the results in terms of year per unit gain. So we want it to be as small as possible. They had different traits with different levels of heritability. And in white, you have the results of the phenotypic selection, and in black, genomic selection, depending on the size of the population. So you see that unless the population is very small, genomic selection is always better than phenotypic selection. So at the end, they went from 14 or to 25 years required to get 1 unit gain to only 11 to 24 years with genomic selection. So that's 4% to 25% increases in terms of rate of genetic gain. There are also empirical results that were obtained. So for example, in this one, we showed the relation, the link between the accuracy of genomic predictions, and the level of relatedness between the training and the test set. So when the two sets are very poorly related, if you are considering, for example, different populations, then prediction accuracy can be 0. And then it increases with greater relationship between the two populations. So to maximize this parameter, a good way to implement genomic predictions is to use full sieves or progenies of the training individuals. Among the different studies, it was also considered the effect of the marker type, marker density, and so on. On this plot, you see the effect of number of markers in terms of prediction accuracy for the bunch production in oil palm crosses. So it shows that a few hundreds, or let's say 1,000 SNPs are enough to achieve the maximum accuracy. And finally, because it's not enough to consider just selection accuracy, we also have to consider the rate of genetic progress. With empirical data, this was also studied. That's what you see here. This is for the bunch production trait again. And here, it was a comparison study where we compared the conventional breeding scheme, actual one, where there is no selection on the FFB before the hybrid progeny test. But now, with genomic selection, we can imagine that before making the progeny test, we could predict the genetic value of the parents, of the hybrid parents for this trait. And in the progeny test, we would only evaluate the most promising ones. So what we found is that with the empirical data, if the breeders had done that, then they would have achieved a higher FFB in the crosses selected at the end of the progeny test. You have here different levels of increases, and it depends on the number of individuals you genotype. So obviously, when you take a larger population of application, you can achieve greater levels of genetic improvement. So we suggest a change in the oil palm breeding scheme. So again, you have here the conventional breeding with 2 cycles. And here we have a modified version where genomic selection is applied at different levels, either before the hybrid progeny test here. So that's the example I gave in the previous slide. So this increases selection intensity because genomic predictions, they can be applied on the whole set of individuals you have in the field and then in the nursery. So that's greater selection intensity. So that's what increases the genetic gain in this suggested breeding scheme. To summarize our results in oil palm, we can say that now with genomic predictions, we can make a pre-selection before the hybrid progeny test and before the clonal trials. So this will allow 2 things. First, the selection on a much larger population of selection candidates, because it's just a matter of cost, and the cost of genotyping either hybrid parents or clones, potential clones, that's lower than the cost of phenotyping in the field. Also, this genomic pre-selection can be applied on more traits. Currently, there is a pre-selection before the trials, but it's only made on the traits, and typically a single trait with high heritability. with genomic predictions, we can do that on all the traits as long as we have a training population for these traits. If we go back to the breeder's equation, what we found is that we achieve an increase in selection intensity with genomic predictions, but also we have a decrease in the selection accuracy with genomic predictions compared to the accuracy of the estimates of the genetic values we make from the field trials, which is around 0.9. So for genomic predictions, it's lower. But the overall result is that the increase in selection intensity is much higher than the decrease in selection accuracy. So at the end, the genetic gain is greater. And for now, we did not consider shortening the length of the breeding cycles, but in future cycles, it will be possible because we maybe consider some cycles without phenotypic evaluation. So that's a possibility for further increase in terms of genetic gain. Here is now the final part of my talk. I will present a few trends, current trends in the field of genomic predictions. The first one I present is the use of multi-environmental data. So the point is to make prediction of the genetic values for new genotypes in known environments. So that's what I presented so far. But also now using multi-environmental trials, the idea is to be able to predict the value of known genotypes in new environments, but also here, the final point, the most challenging one, to predict the performances of new genotypes in new environments. The environment can have complex effects on phenotypes. This is clearly illustrated in this example, which shows different types of phenotypic variation on the y-axis as a function of environmental effects on the x-axis. This type of relationship is called a reaction norm. The three lines or curves shown in each case represent three different genotypes. We therefore see reaction norms with varied profiles that are not necessarily linear. Under these conditions, it is important to be able to select materials suited to the expected range of climate change in production areas, either by identifying stable material or by identifying different materials specifically adapted to particular expected conditions. Various approaches have been developed to incorporate environmental data into models. Thus, reaction norms can be modeled directly using random regression. Here is an example of a result obtained for a tree species larch for a trait expressed as a function of available soil water. In the context of genomic prediction, this can be used to predict not just genetic value for each individual, but a phenotypic response curve as a function of an environmental gradient. Equivalently, one can also use environmental covariates to construct an environmental similarity matrix that quantifies the similarity between the environments in the same way as similarity matrices calculated from SNPs between individuals. Another major area of interest for genomic prediction is the use of other types of information. So far, in what I presented, and in most genomic prediction studies, the information used are the SNP data. But now we can also consider different layers of information that goes between the SNPs and the phenotypes. So first, the SNP information can be grouped into haploblocks that can be more informative. It's also possible to consider structural variants, like copy number variations, insertion deletion, and so on. It can be possible to consider genomic features that can come, for example, from annotation studies or QTL studies, where you have information about the possible function associated to the genes where your SNPs are located. And then you can use these other layers of information ranking from the epigenome to the metabolome, considering also transcriptome and proteome. Another area with a lot of development for genomic predictions is the use of methods taken in the field of artificial intelligence. So artificial intelligence consists in a wide variety of methods, in particular deep learning, but also other approaches. So the idea here is that when we consider multi-environment data, large populations and multi-omics data, then we have a very big data set comprising a lot of heterogeneous data. So data of different types, unbalanced, incomplete, and so on. So this is actually the definition of big data. And the idea is that for, let's say, limited amount of data, deep learning or AI tool could perform similarly compared to conventional statistical methods. But at some point, AI methods should be more performant than the conventional tools we use and the ones that I've presented, like BLUP methodology. If we talk specifically about artificial neural networks, there is a wide variety of methods with varying network architecture. For example, different number of layers and neurons per layer, as well as different types of networks, which can be seen on this slide, and a set of network calibration hyperparameters, such as the learning rate or the function used to measure the difference between predicted and observed values. Neural networks require theoretical knowledge different from that of conventional statistical methods. In machine learning, for example, the knowledge of quantitative genetics is no longer essential. There are also differences in terms of implementation of the method. For machine learning, it's quite different because it involves a lengthy phase of testing architectures and combinations of hyperparameters before arriving at the final model. Finally, I will discuss high-throughput phenotyping. It enables the creation of large training populations at reasonable cost and phenotypic selection, which I will not cover here. High-throughput phenotyping relies on various tools, including culture chamber or phenotyping platforms with automated imaging, and also field-based equipment such as phenomobiles. For high-throughput phenotyping in the fields, unmanned aerial vehicles are also largely used now. You see examples here. This type of data is used in annual crops to estimate the crop canopy height. You have digital elevation model that measures ground elevation plus crop height, and the digital terrain model, which measures ground elevation. And from that, you can obtain canopy height. So that's all for my talk, and I thank you for your attention.